So some of the best ways to assess MRD again are using flow cytometry or using sequencing. Some of the advantages of flow cytometry is that it does require a sample, and it's something that labs can do in academic centers. For example, each lab at each academic center can set up their own assay. This is something that can be learned. But some of the disadvantages of this technique is that it usually requires a lot of time for you to do the assay. So you can do flow cytometry and you can use different colors to detect the myeloma cells, for example, and you can use different tubes. But the more cells that you have to analyze to detect the myeloma cells that are kind of left, the harder it is to do the test. So it could be a test that could be many hours, for example, that is very costly for the department that is doing this test. Whereas, for example, another with sequencing, it's not, there's a price because it's a commercial assay, but it's not something that you have to have, for example, your Department of Pathology do and create an assay and spend 5 or 6 hours doing the test for just one patient. And again, the issue with flow cytometry is that every place is different. So in this country they do it this way, and this other country they do it this way, and then in New York they do it this way, and in Houston they do it this way, and in LA it's this other way. So everyone has a little bit their own assay that they do, and what we just have to be careful is the sensitivity, and I think that's something that people are aware of. And the nice thing with sequencing is that it's DNA sequencing. So the DNA, it's the same almost everywhere. And also there are commercial assays that is the same assay. You just send the sample to a centralized location. They do the test, and of course it's a centralized location. So the test is going to be the same whether you send the sample from New York or Houston or LA or something. So those are like the different tests. But I would say if you are a physician in the community and you don't have access to a very good flow cytometry lab that can look in a lot of cells and stuff, you can just probably send the commercial assay, and that way you can test, you can tell your patient what is the response that they have, and that's something that you just have to draw an extra sample and ship it, right? So that would be my basically recommendation if you don't have access to these labs that do the advanced flow cytometry testing. Mm-hmm.